On the morning of the 19th of March, 2011, a 4-year-old polar bear at the Berlin Zoological Garden turned at the edge of his enclosure pool, suffered a seizure, and fell into the water. He did not come out. There was a crowd watching. His name was Knut, and for a stretch of the late 2000s he had been, by a considerable margin, one of the most photographed animals on Earth. The question that followed was simple enough that a child could ask it, and a great many children did ask it. Why did Knut die? For 4 years, the honest answer, given by the people best placed to give one, was that nobody knew. Then, a neurologist in Berlin went back to what had been kept of Knut's brain and ran a test that had never been run on an animal before. It came back positive. And what it told him was not, in the end, really about Knut at all. It is worth being precise about what that test was, because a good deal of what circulates about this case is wrong. Knut's genome was never sequenced. Nothing was read letter by letter, no ancestry was traced, and at no point was there any question about what species he belonged to. What was tested was his cerebrospinal fluid, the clear liquid that surrounds the brain and the spinal cord, along with thin sections of preserved brain tissue, assessed against diagnostic criteria that had been written for human patients in hospitals. The genetics in the paper comes down to a single comparison. The researchers took the stretch of protein that the human test is built to detect, lined it up against the equivalent stretch in a polar bear, and confirmed that the two were similar enough for the comparison to mean anything. They were. Which is a far smaller claim than a genome, and a considerably stranger one. Knut was born in the Berlin Zoological Garden in December of 2006, one of 2 cubs, and he was rejected by his mother within days of his birth. His brother did not survive the first week. Knut was taken in by the zoo's staff and raised by hand, principally by a keeper named Thomas Dorflein, and he became the first polar bear cub born at that zoo to survive infancy in more than 30 years. He was introduced to the public in March of 2007, at 3 months old, small enough to be carried, and the response was immediate and entirely disproportionate. He appeared in daily presentations alongside his keeper. Visitors arrived from other continents specifically to stand in front of his enclosure. Merchandise, television coverage, magazine covers. The scientific paper that eventually explained his death records the commercial side of this in a single sentence, and it is a startling one. Knut's popularity produced the most profitable period in the zoo's 163-year history. He grew. The daily presentations stopped, as they were always going to, and he was moved in with 3 adult females. He was 4 years and 3 months old when he died, against a life expectancy for his species of roughly 30 years, which is to say that he lived somewhere near 1/7 of a polar bear's life. If this is holding your attention, subscribing helps more of these reach the people who'd want them. The investigation that followed was not casual. It was led by the Leibniz Institute for Zoo and Wildlife Research in Berlin, and it was thorough to a degree that is unusual for any animal, let alone a zoo animal. The necropsy established quickly that Knut's seizures had been caused by encephalitis, inflammation of the brain. That much was never in doubt. The question was what had caused the inflammation, and the assumption, entirely reasonable at the time, was that something had infected him. Brain inflammation in a captive animal means a pathogen until proven otherwise, so they went looking for one. They screened for viruses, for bacteria, for parasites, for anything that had ever been documented as causing encephalitis in a carnivore. And when the known candidates came back negative, they went looking for unknown ones. That search became its own piece of published work. In 2014, Sentix and colleagues described, in the Journal of Comparative Pathology, a comprehensive next-generation pathogen analysis pipeline built for captive and free-living wildlife. A system for finding infectious agents nobody has catalogued yet. It was built, in large part, because of one bear. A related line of work characterized a previously undescribed group of endogenous retroviruses in polar bears and giant pandas, published in Virology in 2013. All of it came back the same way. No pathogen. The file entry that stood at the end of 4 years of work was encephalitis of unknown etiology, a formal way of saying that the brain was inflamed and that nobody could say why. And here is the part that matters, because it explains why this took as long as it did, and it is not a failure on anyone's part. The alternative explanation, that a body can produce antibodies that attack its own brain with no infection involved at all, had only entered the medical literature in 2007. Dalmau and colleagues had described it in humans that year in the Annals of Neurology. Knut died 4 years later. Nobody had a reason to go looking for a brand new human disease in a polar bear because nothing had ever suggested that an animal could get it. What changed was that one of the people who had spent those years working on the human version happened to read the phrase on Knut's file. Harald Preuss is a neurologist at the Charité in Berlin and at the German Center for Neurodegenerative Diseases. In 2010, he had published a retrospective study in the journal Neurology, going back through human patients who had been diagnosed with encephalitis of unknown origin and testing their stored samples for antibodies against the NMDA receptor. A meaningful number of those undiagnosed patients turned out to have had the disease all along. So the phrase on Knut's file, encephalitis of unknown etiology, was, to Preuss specifically, not a dead end. It was a category he had already learned to distrust. The mechanism is worth spelling out because it is genuinely unintuitive. The NMDA receptor sits on the surface of nerve cells throughout the brain and is central to how those cells signal to one another. In this disease, the immune system produces antibodies against one particular part of that receptor, the NR1 subunit, and those antibodies bind to it and interfere with it. The brain is not being eaten by an infection. It is being switched off, receptor by receptor, by the body's own defenses. In human patients, the result is seizures, hallucinations, memory failure, and in severe cases, dementia. It is now recognized as the most common non-infectious encephalitis in humans. None of which would have been testable at all if not for a decision made in 2011 by people who had no idea what they were preserving material for. When the original investigation closed with no answer, Knut's samples were not disposed of. Fluid and sections of brain tissue on slides went into storage, the standard practice for an unresolved case, and the reason there was anything left to examine 4 years later. The entire 2015 result rests on a freezer in Berlin, and on the professional instinct that says you keep the material when you cannot explain the death. Preuss, working with Alexander Greenwood and colleagues at the Leibniz Institute, tested Knut's preserved cerebrospinal fluid for those antibodies. They were there at high concentration, specific to the NR1 subunit. Then they went to the tissue. Under the microscope, the pattern in Knut's brain matched what is seen in human cases with considerable precision. Infiltration by plasma cells, the antibody-producing cells of the immune system, with only minimal loss of neurons. That combination is the signature. An infection destroys tissue. This does not. It interferes with it, which is exactly why the disease is treatable in people, and exactly why 4 years of pathogen screening found nothing to find. The authors were careful in how they stated it. They did not announce a new bear disease. They wrote that Knut's encephalitis was almost identical to anti-nmda receptor encephalitis, assessed by the same criteria used for human patients. The hedge is doing real work, and it is the correct hedge. It is worth sitting with how narrow the margin was. There was no systematic review that would have caught this. No procedure existed that sends unexplained animal encephalitis for autoimmune testing, because until this paper, there was no reason for such a procedure to exist. The case turned on one specialist in one city reading one file and recognizing a phrase from his own research. Had the samples been discarded, or had Preuss been working somewhere other than Berlin, Knut's cause of death would simply have remained unknown, permanently. And the first recorded case of this disease in an animal would still be sitting in an archive waiting for someone to think of it. This next part is the reason the case is still cited. Subscribe if you'd like more of these. Knut is the first non-human animal in which this disease has ever been demonstrated. Not the first bear, the first animal of any kind, wild, captive, or domestic. And the implication the authors draw from that is much larger than one polar bear in Berlin. If a disease that was only described in humans in 2007 was present in a zoo animal in 2011, then it was almost certainly present in animals before 2007, and it is almost certainly present in animals now. It has simply never been looked for. Their conclusion is that anti-nmda receptor encephalitis may be a disease of broad relevance to mammals that has, until now, gone undiagnosed. And, more broadly still, that errant immune responses may sit behind brain disease considerably more often than has been assumed. There is a practical edge to this. In humans, the disease responds to treatment. The immune response can be suppressed and patients can recover, sometimes substantially. An animal presenting with unexplained seizures has, historically, been worked up for infection and for tumors. This paper says there is a third thing to test for, and that the test already exists, and that it works on tissue from a species separated from us by tens of millions of years of evolution, because the part of the receptor the antibodies attack is conserved across mammals closely enough that the human assay reads it. It is worth pausing on the pathogen pipeline, because the way it is usually told gets it backwards. 4 years of screening for infectious agents in Knut's brain produced no answer. That looks like wasted effort, and it was not. The exhaustiveness of that search is precisely what made the second look possible. If the pathogen work had been cursory, an autoimmune explanation would have remained one possibility among many, and the antibody result would have been suggestive rather than persuasive. Because the infection hypothesis had been pursued as far as the available technology could pursue it, and had failed, the field was clear. The null result was the foundation. That pipeline also outlived the case that produced it. It was published as a method for wildlife generally, captive and free living, and it is available to anyone investigating an unexplained death in an animal, which was never the point of it and is arguably the most useful thing to come out of the whole affair. What remains true about Knut, once the tension is taken out of it, is fairly plain. He was born in a zoo in Berlin in December of 2006. His mother rejected him. He was raised by people, which is not a normal way for a polar bear to begin, and he was the first cub at that zoo to survive infancy in 3 decades. He was extremely famous, for reasons that had almost nothing to do with him. He lived 4 years and 3 months. He developed a disease that nobody knew an animal could develop, and which, in a person, a doctor would have known how to treat. He is still in Berlin. His preserved form stands at the Museum für Naturkunde, and the museum is firm about the distinction. He was not stuffed in the conventional sense. What is on display is a form built to his exact proportions, covered with his own fur, finished with glass eyes. In 2013, it traveled to the Netherlands, where more than 150,000 people came to see it. What is not known is how many other animals have died of this. Nobody is counted, because outside of this one case, almost nobody is testing. The paper does not close that question, it opens it. And the answer, when it finally came, did not come from reading Knut's genome. It came from someone reading his file and recognizing a phrase.